Today we are going to discuss how the regulation of genes takes place, how the regulation of expression of the genes takes place in prokaryotes with the help of Lac OPERON model. I am Dr. Rajeev Basumatary from DR College Golaghat. So let's start. So what is gene expression? So the gene expression is a process by which the informations which are located or which are encoded in the gene in the form of base pairs of DNA sequence is made into a functional gene product such as protein or RNA. So the gene expression is actually the process by which the genes form the proteins. So as we all know, the process of formation of protein it includes two steps: transcription and translation. So the transcription is actually the copying of an RNA molecule from DNA or the gene, and from this RNA molecule the sequence in the RNA or mRNA is then translated in the form of sequence of amino acids, which we all know as the polypeptide chain. And this polypeptide chains then go inside endoplasmic reticulum and some in mitochondria. And inside endoplasmic reticulum and mitochondria, these proteins are modified to form, these polypeptide chains are modified to form the functional protein. Now let us understand why the gene expression need to be regulated and why the process of protein synthesis need to be regulated. There are actually various reasons why regulation is required by the cell. Because if the cells continue to form protein in an unregulated way, so there is investment of very large amount of energy and also the time. So that is why the cells, they turn on or they start transcribing a gene only when their product is needed, and when the product or that protein is not required by the cell, at the time the genes just are switched off. Moreover, if a cell continue to form protein in an uncontrolled manner, there will be stacks of proteins in the cell, and which will prove to be lethal for the cell in the long run. So that is why the regulation is very important process for a cell, and if the protein synthesis or gene expression is not regulated, it will be a lethal consequence for the cell. The gene expression is regulated in various steps and is different in prokaryotes and eukaryotes. So in eukaryotes, the process is very complicated and it is regulated in multiple steps. That is, before transcription, after translation, after transcription, after translation, before translation, etc., or even during the modification of the different polypeptides. But in case of prokaryotes, the process is simple, and the gene expression is regulated only during transcription or just before transcription. So this process we are going to discuss with the help of Lac OPERON model. So to understand Lac OPERON, we have to know what is operon. So the operon is actually a set of genes that are transcribed under the control of a single set of regulatory gene, and they are transcribed together as a single polycistronic mRNA. That means the operon is actually a group of genes, means it is a group of 2, 3 or 5 genes which are actually they are transcribed into a single mRNA molecule, and that mRNA molecule will contain the sequence of all the genes, and this type of mRNA molecules are known as polycistronic mRNA. And the genes, this type of genes which are transcribed in grouped into a single polycistronic mRNA are known as operon. And one more important thing is that such operons are regulated or are controlled by a single set of regulatory genes. So there are two types of operons. They are inducible operon and repressible operon. The inducible operons are those which are generally inactive or remain switched off, and they start transcribing, or the genes are switched on only when the substrate to be metabolized is present or is available in the cell. So the example is Lac OPERON, which we are going to discuss. And the another operon is called repressible operon, and it is just opposite to inducible. That means in this system the operon is usually in on mode, so that the transcription occurs normally to synthesize the enzymes leading to synthesis of its product. But the operon is switched off when the product or the metabolite becomes available. Example is tryptophan operon. Now let us discuss in detail about the Lac operon. So the Lac operon stands for lactose operon. Lac stands for lactose and is associated with the transport and the metabolism of lactose in humans and many other enteric bacterial cells. The Lac operon is actually composed of two main components. There are two sets of genes which are known as regulatory genes and structural genes. The regulatory genes are those which are responsible for regulation of the genes which are actually going to transcribe into mRNA. And the regulatory genes are, there are three types of regulatory genes which are present in the regulatory gene set. They are known as inducer gene, promoters, and then operators. On the other hand, the structural genes are the genes from which the mRNA is transcribed. And the structural gene, in case of Lac operon, there are three structural genes are present. They are Lac Z, Lac Y, and Lac A gene. So this is the diagram of a Lac operon, where Lac I means inducer, then promoter, and then operator. Altogether they are regulatory genes which is going to regulate the structural genes. Structural genes are Lac Z, Lac Y, and the Lac A. The structural gene Lac Z, it forms into a product which is known as, it's actually an enzyme which is known as beta galactosidase. Then Lac Y, it forms galactoside permease, and the Lac A, it forms the beta galactoside transacetylase. So all these three enzymes together, they are responsible for metabolism of lactose by the bacterial cell. So that is why this operon is known as Lac operon. So the Lac operon, or the three structural genes, they are first transcribed in the transcription process with the help of RNA polymerase enzyme. And in this transcription, all the three genes, they will be transcribed into a single mRNA molecule, and this mRNA molecule, it will contain sequence of Lac Z, Lac Y, and Lac A. And such type of mRNA, they are known as polycistronic mRNA. And after that, this polycistronic mRNA will be splitted according to the sequence of the structural genes, and they will undergo the process of translation separately. And the sequence of Lac Z, it will finally give rise to beta galactosidase enzyme. Then Lac Y, it will form permease, and then Lac A, it will form beta galactoside transacetylase enzyme. If we look at the functions of the three enzymes, the beta galactosidase, it has two different functions in the organisms. First, it can go through a process called transgalactosylation to make allolactose, creating a positive feedback loop for the production of beta galactoside. Second, it can also hydrolyze lactose into galactose and glucose, which will proceed in glycolysis. Next enzyme is beta galactoside transacetylase. It is an enzyme that transfers an acetyl group from acetyl coenzyme A to beta galactoside. Then the third enzyme, galactoside permease. It is a transmembrane transporter enzyme or protein that pumps the beta galactosides, including the lactose, into the cell using a proton gradient in the same direction. The permease increases the permeability of the cell to the beta galactoside. Now let us understand the three statements about the Lac OPERON. First, the Lac OPERON is switched on or transcribed only when there is lactose in and around the bacterial cell to make the three enzymes which we have just discussed. Second, the enzymes that are produced by the Lac OPERON, they will help the bacteria to metabolize or the use or help in the use of lactose. Third, when the lactose is fully utilized and no lactose are left around then the Lac OPERON will automatically switched off. But how the Lac OPERON will switch on in the presence of lactose, and it will switch off when the lactoses are fully utilized. So to understand this process, we can look at this diagram. The Lac OPERON works differently in two situations, that is in the absence of lactose and in the presence of lactose. So when there is no lactose around the cell or in the cell, or when there is absence of lactose, in such situation there is no need of formation of the enzymes that break down the lactose or that helps in the metabolism of lactose. So that is why in the absence of lactose, the Lac OPERON or the genes has to be switched off, or there is no need of transcription to carry out. So to stop the transcription, the three regulatory genes, they work together. So first of all, the regulatory gene, it forms an mRNA molecule, and this mRNA molecule will form a protein which is known as repressor protein, and this repressor protein is active repressor protein, and it has affinity to the operator, that is the third gene in the regulatory set. And when repressor is bound with the operator, the RNA polymerase enzyme, which is at the position of promoter, it cannot move to the structural genes, and when the RNA polymerase cannot move to the structural gene, there is no transcription. That means since there is no lactose in the environment, so there is no need of transcription to carry out and to form the enzymes to use the lactose. So this is one phase of regulation. And the situation is different when the lactose is present around. What happens when lactose is present? This lactose has the affinity with the repressor proteins, which was formed by the inducer gene. That is the first one. And this lactose binds with the repressor protein. As a result, the repressor cannot bind with the operator, and it cannot block the way of the RNA polymerase enzyme. So in such situation, that means in the presence of lactose, this lactose will bind with the repressor protein, and it will make a way for the RNA polymerase enzyme, and the RNA polymerase enzyme will walk around the structural genes like Z, like Y, and like A, and it will carry on the process of transcription, and as a result, a polycystonic mRNA molecule is formed, and from this mRNA molecule, the enzymes beta galactosidase, permease, and trans acetylase will be translated. So what happens? So in the presence of lactose, the enzymes are transcribed, they are translated, and the genes are transcribed, translated, and then the enzymes are formed. So when the enzymes are formed, these enzymes in turn will act on the lactose, and they will help in the metabolism of lactose, and finally all the lactose which are present in the cell, they will be utilized and they will be broken down. And when all the lactose is broken down in the cell, and at that condition, this repressor protein, it will become free again, and it will go and bind with the operator gene, and the RNA polymerase is blocked. So it's a feedback kind of mechanism. When the repressor is when the lactose is present, it will bind with the repressor, and the transcription will take place. When the lactose is absent, the repressor will be free and it will block the RNA polymerase enzyme, and in that way the Lac OPERON is regulated. Okay, for better understanding the process of regulation of gene expression, let us try to explain. Let me try to explain in this whiteboard. So this is the gene, and these three are the structural genes: leg Z, leg Y, and leg A. So these are structural genes, and these three are the regulatory genes. So the regulatory genes are inducer, promoter, and then operator. So what happens when there is no lactose? When there is no lactose in the cell, this inducer, it forms mRNA first, and this mRNA is then translated into a protein which is called repressor. So this repressor protein, it is called repressor because it can repress the process of transcription. How? So you just see. So this repressor protein, there has the affinity to bind with this section of DNA, this section of the regulatory gene, which is known as the operator. So this repressor, they can just go and bind with the operator part of the regulatory gene. So when repressor is bound with the operator, so what happens? Here is one enzyme bound with the promoter region of the regulatory gene, and this enzyme is known as the main enzyme, RNA polymerase. So RNA polymerase is the enzyme which is responsible for copying of a mRNA molecule from this DNA, or the process of transcription which you call. So RNA polymerase is responsible for transcription of RNA from DNA, or the copying of RNA from DNA. But when the repressor is bound with the operator, then the RNA polymerase cannot go this way, cannot work in the DNA, and so it cannot work. So there is no transcription, or there is no formation of mRNA. So when there is no mRNA formation or no transcription, then there is no formation of the enzymes of this leg Z, leg Y, and leg A. Those three enzymes, beta galactosidase, galactoside permease, and transacetylase, these three enzymes will not be formed since there is no transcription. And why there is no transcription? Because the repressor is bound with the operator and it is blocking the way of RNA polymerase. So the summary of the story is that when there is no lactose, repressor, it goes and binds with the operator, RNA polymerase is blocked, and as a result the transcription process is stopped and there is no formation of enzyme. So no lactose, no enzyme formation. The story is different when the lactose is present. When the lactose is present, the whole story goes in an opposite way. What happens? The repressor protein that was formed by the inducer, this repressor protein now binds with the lactose instead of the operator. So repressor will bind with the lactose when the lactose is present. If there is no lactose, then repressor will bind with the operator. But when lactose is present, instead of operator, the repressor protein, it will go and bind with the lactose. So when repressor is bound with the lactose, when the repressor are engaged with the lactose, what happens? This RNA polymerase, which is in the promoter, this RNA polymerase is now free. It is now free to move. It can walk in the structural gene, and as a result, this RNA polymerase will copy this structural genes into a long mRNA molecule. This mRNA molecule has the sequence of these three genes, Lac Z, Lac Y, and Lac A. So this type of RNA molecule carrying the sequence of more than one gene is known as polycistronic mRNA. 5 prime, 3 prime. So this polycistronic mRNA is formed, and from this polycistronic mRNA, the three enzymes will be translated separately. So this is number 1 enzyme, number 2 enzyme, number 3 enzyme. Number 1, number 2, number 3. Beta galactosidase, galactoside permease, and transacetylase. When these three enzymes are formed, these enzymes will go and act on the lactose available in the cell. As a result, after some time, all the lactose will be utilized, or they will be metabolized. They will be broken down, and as a result, there will be no lactose left in the cell. So when all the lactose are utilized, when the cell becomes lactose free, the repressor molecule, it becomes free, and when there is no lactose, the repressor is now free to bind with the operator. The repressor will go and bind with the operator. So when the repressor will bind with operator, the RNA polymerase will again block now. So as a result, from this point, there will be no transcription and there will be no formation of the enzyme. So the whole summary is that when lactose is present, the enzymes will be formed, and due to the formation of enzyme, the available lactose will be utilized. When all the available lactose is utilized, the enzyme formation or the process of transcription will again stopped. So in this way, the presence of lactose will induce the Lac OPERON to form its enzymes, and the absence of lactose, it will block or it will suppress the function of the Lac OPERON. So this is all about Lac OPERON. Thank you.